to today's Strategic Farming Let's Talk Crops program. We're happy to have you joining us today on our today's session of emerging corn and soybean diseases. And these sessions are brought to you by University of Minnesota Extension with generous support from the Minnesota Soybean Research and Promotion Council and the Minnesota Corn Research and Promotion Council. I'm Claire LeCan, Extension Educator in Crops, and I'm stationed in our Andover Regional Office. And we welcome Dean Malvick, who's our University of Minnesota pathologist with Extension. So with that, I will turn it over to our main event here, Dr. Dean Malvick.
All right, thank you, Claire. Good morning, everyone. Glad to be here. Just to start out with, the question of this topic came up of emerging corn and soybean diseases. And when we think carefully about the words, they're emerging in some ways, and many ways they're not. And what I mean by that is most of these diseases that we know about already, they've already emerged, right? They're already here in Minnesota. We don't know exactly what might be on the horizon, although we can speculate. Some of the diseases that have come into Minnesota, certainly they weren't expected, long before they came. For example, tar spot, which we'll touch on today. Nobody thought about that even being in the US before 2015. So the question always is, what is on the horizon? And, there are folks looking out there. And I know I've seen lists of things that might be coming, other diseases that could be very important, but, they haven't come, but others have. And that's kind of what we'll focus on today, the ones that we know about. And again, in choosing topics for today, that was the theme, is emerging. There are also new emerging questions and possibilities to address some of these diseases. And the other thing is, as Claire mentioned, people send in questions. And some of you in the audience sent in a nice number of questions, a few of you. So I actually added a few topics today specifically because those questions were sent in ahead of time. And as always, there are a lot of things we could cover that we won't today. And that's one reason why we have time for questions at the end. So let's get right into this. So we'll just go in disease by disease. Again, there was a question about gray leaf spot. That is not a new disease in Minnesota, I think, as we all know. But it has not been an especially important one as compared to states to the south of us or the east of us especially. And one of the challenges with this is, diagnosis, a little bit at least. That bottom picture in the black border shows the classical gray leaf spot symptoms with those gray rectangular lesions bordered by the veins. But you see all the other pictures there too, they're also gray leaf spot, and they can look different. And as I said, we don't know of many instances where this has been really important. There have been scattered fields here and there, but it is common. It's very common in the fall in a lot of fields across southern Minnesota at low levels. And so why don't we see more of it? This is a fungal disease, and we don't typically see that prolonged high humidity that really is favorable for this disease. Like when I've seen it at really, really high levels, say in southern Illinois, this kind of weather is typical down there in mid to late summer, even the middle of central Illinois, much more so than here. Some summers we do have very high humidity, and some protected fields, low areas, valleys, certainly we can have these conditions, but we tend not to have them on a broad scale. And this map was made a long time ago about, the highest risk areas. What's interesting about it, so the green shows the highest risk, and the yellow, which doesn't show up very well in the legend on the bottom, but shows more of a moderate risk. And that still seems to match pretty well what we see, even decades later, that this was made. So it does reflect that certainly that disease does have potential to be a problem here. And if we have wet, humid summers, which we certainly haven't had recently, this could be an increasing problem. And the ways we won't think about this, managing this if and when we have problems, certainly there are resistant hybrids. The seed companies diligently work on, developing and maintaining resistance to this. And fungicides are also quite effective. So those are the two approaches we can take if and when we have a problem. So I'm going to just go through these, rather quickly. Again, bring up questions if you have them. I certainly won't answer all the questions you might have about these. Now, northern corn leaf blight, another leaf disease on corn, which we've seen at actually at higher levels than we've ever seen. Usually, I would say we see this at we've seen this at higher levels than we've seen gray leaf spot. So here are a couple of pictures showing you these canoe-shaped lesions that often may be an inch to up to 4 inches long. And to find single lesions like this on a scattered leaves in the field are not uncommon. In fact, they're easy to find most summers. What we don't see very often is levels like we see in that picture on the right. That happens to be from Canada, but it does happen here too. And what conditions favor this disease? This prefers cooler conditions and also wet. All these other things, cool dew periods that are at least 5 hours. And again, it shows how long it can take for spores to germinate and for infection to occur. And that was one of the questions that was submitted ahead of time. What is kind of the latent period, the time between when a pathogen lands on a plant or contacts a plant and when we actually see symptoms, or when we see actually entry into the plant and infection. That all makes a big difference in terms of our fungicide timing. And for every disease, it's a little bit different. For this one, like you can see here, we can get infection. The fungus can enter the plant, within, 4 to 8 hours after that spore lands on a leaf, potentially. Now, it would be some days after that before we see symptoms, but things can happen fairly quickly in that early stage, especially. And then managing this, certainly we have resistant hybrids, tillage and crop rotation, anything that reduces that residue and the amount of pathogen that's out there on infected leaves. And then, of course, fungicide applications as well. I'm not giving you specific fungicide recommendations or thoughts on efficacy, but I'll mention this right up front right now, that for those of you that are looking for that kind of information, how different fungicides affect or how effective they are for these different pathogens and diseases, the Crop Protection Network is an excellent source. And if you saw Strategic Farming last week, you saw a presentation on that and how those data are arrived at, how they're acquired, and how they're summarized by Kirsten Wise from Kentucky. She's one of the coordinators behind that. And there's a lot of very good information on that Crop Protection Network website, including fungicide efficacy evaluation information. And another question that came up ahead of today's session was stalk rots. And there's nothing new and emerging about these. These have been here and been problematic for a long time. And I'm sure most of you have seen them at one level or another in fields, often at fairly high and severe levels. And what I list here are the primary types of stalk rots we see in Minnesota. Anthracnose is especially common in many areas. Gibberella and Fusarium, and then more recently, Physoderma has been seen more often. And then just to remind you what these look like, anthracnose is the easiest one to diagnose because it results in this dark black discoloration in the outer rind of the drying stalk. Typically late in the season, you see it more clearly. And you can see how completely it can rot the inside of the stalk, of the pith area, in that picture on the right. Gibberella stalk rot. This is another fungal disease. It sometimes has this pink kind of discoloration associated with it. And it's a reminder it's the same pathogen that causes Fusarium head blight. So that's why rotation of wheat or small grains with corn can be problematic because it can increase that inoculum load and result in a bigger problem for both. And Fusarium stalk rot is also quite common. It's not always easy to diagnose, but it's one of the very common pathogens and diseases out there. And then again, I mentioned Physoderma. That's been seen more commonly in our region, in the last 6 to 10 years, especially. It can cause issues on the leaves and this dark black discoloration, especially at the node, resulting in breakage right at that point sometimes. And every year there's a group of folks that go out around the state, do surveys, try to see how much stalk rot is out there. I just picked 2 samples from a few years ago, again, showing, the red and the yellow dots, especially showing, between 25 and 100% stalk rot in the fields. You can get an idea of how widespread it is here in Minnesota. It's everywhere corn is grown, essentially, is the main point of this. And like any disease, it'll vary from year to year about how much is actually out there and where and when it occurs. And of course, when it occurs early is when it's perhaps the biggest problem because it can actually take out yield. We usually don't see that. The bigger problem is, lodging early, partially due to, wind damage and weakened stalks, and then resulting harvest challenges, etc. How do we manage this? Well, there's no simple formula for this one, partly because there are many different pathogens. And the number one thing to do is reduce stress on corn plants. Minimize, I guess, leaf disease too. I mean, that's a key thing. So, and the point there is if we reduce the leaf area available for filling the ear in terms of photosynthesis, we also reduce the amount of photosynthate and energy that can go into the stalk that can defend itself against these stalk rot pathogens. That's kind of a common idea that follows behind this. Anything that stresses the plant, reduces photosynthesis, can increase stalk rots. Then, as many of you seen, drought can be part of this too. So it's a complex problem, widespread at times. Sometimes we don't see it. Sometimes it's very severe. In high plant populations can make it worse, early maturation, and of course, drought, like I said. Now, in terms of a truly emerging disease, tar spot of corn. We've been talking about this for a few years. I just want to raise a couple of points. Some of the questions that came up, especially was, is it spreading and where is it spreading? This map is available on a website called the Corn IPM PIPE. And PIPE stands for Pest Information Platform for Extension. Okay, so you can just search it up on Google or whatever, and you can find this map. And it's updated throughout the season and will show where tar spot has been found. So this will start being updated sometime in July, very likely, when the disease starts appearing in the Midwest. And you can see, relatively live updates, at least week by week, where the disease is occurring and being found. This map shows the overall summary of the past 5, 6 years, 7 years, actually 8 years now, of where tar spot has been found. In total, the colored counties in total show all the counties where tar spot has been detected. The yellow gold counties show those counties where it was detected in 2023. Part of this reflects many of the areas that were very dry. Just as a reminder, the disease was first found in late 2015 in north central Illinois and Indiana. It spread out from then over the past 8 years. Now, the big question is it spreading? And where does it really occur? Now, it's definitely been spreading, west into Nebraska and Kansas and into Missouri. That's been very clear the last few years. It's been spreading more on the East Coast, more in Pennsylvania, New York. In Minnesota, we've had very dry conditions across the central part of the state, as you all know, the last 2 years, which doesn't favor really development of the disease at the levels of dryness we've had. So the gray counties show where it has been found, but I wouldn't be surprised at all if it was actually further west and maybe even north of that and just not detected at very, very low levels. And so this is part of the issue. And again, the map that I show here shows Minnesota counties where it has been found. And this is from 2022. So that's why the color is a little different, because it shows the counties where it was that year. And that orange arc in the southeast and the star shows the area where the disease has been by far the most important. If you go west, as we go west and north of there, the disease has typically been at low to very low levels. And what I'm talking about there is, sometimes single to just a few tar spots on a leaf, maybe on a plant, maybe on 10 plants. So not easy to find, not easy to see unless we're deliberately looking for it. And even then, it may be very hard to find. So that's why I say it's hard to know exactly how far west and north the disease has been. And if it's further north and west, it has had no consequence on the crop whatsoever. But it does indicate that the disease is spreading wherever these counties are, where it's been found and where it is, and where the risk is greatest. Now, the question I think a lot of people have is that what are the conditions that really favor this disease? And I think we've been learning a lot of information, and not to get into this too deeply, but 2 key points. We thought it was the disease that needed a lot of water and really cool conditions. And we've seen that's not always true. And for example, cool conditions, I know, for example, in central Illinois, there have been problems with it in past years where it's been typically very warm central Illinois conditions. So it doesn't always need cool weather, although cool weather seems to help it. And then what about moisture? Now we know we didn't see much of this in 2023, I think partly because we had very dry conditions. But let me say this, we did an inoculation trial here in St. Paul with a graduate student, Jose Solorzano, and he inoculated corn plants in roughly the third week of July, and it was fairly dry here. It rained a little bit after, but very little. And he got very nice infection developing on the plants. So the tar spots were visible 19 days after inoculation. It spread from those initial about that amount of time later. So the question came up was, again, what is the latent period? And how do we think about this in terms of management? So we can have, again, infection and disease development occurring over this 19-day period before we actually see symptoms. As a reminder, most fungicides really work best to prevent or slow down the initial infection and growth inside the plant. They're not as good at all at curative, although some have some potential to do that. They're most effective when you can get them on ahead of any substantial infection. So that's part of the challenge with this, right? If we wait until a lot of infection is occurring, maybe even before we have much in terms of symptom development, we'll probably lose some of the efficacy of our fungicides. Now, a little bit more about the temperature conditions. We found that we could get infection at moderate temperatures, low humidity, very little rain. And a larger study was done looking at conditions across the Midwest where tar spot did and did not develop. And there's a whole paper published on this, but two key points. Temperature is the crucial element, the most important environmental variable that they found that influenced tar spot development. Again, cool temperatures seem to be most favorable. Again, below 73 Fahrenheit in their information. Now, like I said, we can get tar spot developing in higher temperatures, but maybe the nighttime temperatures when they're cooler is enough to keep it going. Still a lot we have to learn. What about moisture? At one point, we thought maybe this was something like white mold. It really needs a lot of moisture to keep going. Doesn't seem to be the case. This can do well, and maybe even does better in drier conditions than that. Still needs moisture with how little we saw this year with no moisture in many places. But the point is here, moisture was lower. So extended periods of high humidity, especially at high temperatures, seem to actually hinder the progression and development of tar spot. So I think this raises a lot of questions, but it does show that cool temperatures are favorable, which is something we knew, but we don't need as much moisture probably as we thought we did. And managing this, again, this is nothing new here. More and more hybrids are being developed and marketed and rated for different levels of susceptibility. And so choosing the least susceptible hybrids can make a big difference. It's been shown to be very clear in some of the areas that this is especially important. Scout fields to try to figure out where and when it's developing. Mostly we see this disease in mid to late August, but it can develop much earlier, and fungicides can be quite effective. And this timing of the tasseling to R3 seems to be the optimal timing. We go much after R3, we lose efficacy and ability to stop the epidemic. And again, the crop protection network. Here's an example of a fungicide efficacy table for corn leaf diseases. And this highlighted column here, and that shows the tar spot ratings. And the fungicides rated, from very good to good, etc., for tar spot management. And the good thing about this is we have many different fungicides that are good to very good for this particular disease. And the last disease I want to mention for corn is southern rust. In terms of an emerging disease, this is definitely one we haven't seen much the last 2 years because of, I think, the dry conditions also in many places. But we do see this, and this is a concern because this is a potential bigger problem than common rust. So something to watch out for, especially late in the season when the weather gets really warm. And now moving on to soybean diseases and some of the questions that were brought up. Okay, first of all, here's another I think we could truly call an emerging disease. Frogeye leaf spot fungal disease was uncommon in Minnesota prior to 2017-18, somewhere in that period. And since that time, we've seen increased amounts of this. The exception would have been the last 2 years when it's been so dry. And it has reached levels of concern to the point where fungicide applications have gone on to try to stop this. Okay, it causes these spots on leaves. They're brown to tan spots surrounded by a brown border. That's how we can recognize this. So yield losses can be substantial. Now, we're not seeing this level, 30% in Minnesota for sure, but we've seen it at significant levels that are concerning, like I said. But down south, in southern half of the US especially, this is a significant problem. It's favored by warm and humid weather. There's no question about that for this one. You can manage it with crop rotation, tillage, fungicides. And one of the things we need to keep in mind, and this is a key point, that the pathogen across the US, including across Minnesota, is resistant to the QOI, the strobilurin fungicides. So any fungicide we use has to either be a non-QOI, say such as a triazole or an SDHI type ingredient, or if it's a mixture, it has to have a high enough quantity of those other ingredients. And there's some mixtures out there that don't have enough of the other AI active ingredient to stop this. And actually, you can, again, look at the crop protection network. There's a table like I just showed you. And this one's for soybean diseases, and we'll show you what seems to be and has been shown to be most effective for frogeye. And this just shows an example. We've collected samples. A frogeye leaf spot across these counties doesn't show the whole distribution, but it only shows where we gotten samples. We tested them for this QOI strobilurin resistance, and we found that the population of the pathogen is resistant in all these counties. So I suspect it's resistant in most fields where it occurs across Minnesota. Moving along, wasn't planning to talk about white mold, but there were a couple of questions about it. So very briefly, I want to touch on this. Obviously, there's nothing new and emerging about white mold. It's been here. It's been a problem for decades and decades. I think everybody probably knows how to recognize this. And a couple of things to keep in mind. This really does like cool temperatures, below 75 degrees, during flowering. And it does need wet conditions, long, frequent rains with the soil saturated beneath the soybean canopy. Boggy, misty days are just ideal for white mold. Now, we don't need all of that, but that's the ideal condition. Sometimes just very timely rains in mid-July, when the canopy is full and the temperatures are cool, are enough. And so that really brings out the importance of understanding what we need to do to manage this. Anything we can do to reduce the density of the canopy can be helpful. But also we don't want to go too far, right? And then we lose yields. What else? The other thing here is, the point is, we get a lot of infection early, right after the soybeans start flowering and the canopy and the rows are filling. So that's the optimal time to get a fungicide application on. And now a lot of literature says, at R1, but if we use a strict definition of R1 first flowering, that is, in my opinion, too early. Late R1, maybe even R2, if it's for a single application, would probably be the best timing. Again, based on weather and crop conditions, if we were in the middle of a drought and it doesn't look like it's going to rain, we don't really have to worry about white mold in most circumstances. But of course, we don't know when it's going to rain, so that's always the open question. To keep moving along here, a question came up about soybean cyst nematode. Now we know it's been in Minnesota since the 1970s, but these two maps show that it is still emerging, especially into northern Minnesota. Now it's been found in most counties where soybean is grown, and it's certainly spreading, west into other states and areas there too. But the question that came up here, and this is the question: is there any university data on differences between Alivo and Saltro for SCN control? Now, I think maybe you all know Alivo and Saltro were really developed to control sudden death syndrome. They're excellent products for controlling SDS. That's a fungal disease. They also have effects on nematodes. So they're not a true nematicide, but they clearly can have effects on nematodes, and there is data out there that shows that they can. So to answer this question, I brought up some information from a very newly published paper that was published in January 2024. Now, if you go to this particular URL, this website, what you'll get is the abstract. And that doesn't show the whole paper. The whole paper is not publicly accessible at this point. But what this shows, and the objectives of the study were to evaluate a number of nematode protectant seed treatments for the effect on early and full season SCN reproduction, foliar symptoms and root rot caused by SDS and soybean yield. Field trials were conducted at 13 states and 1 Canadian province over 3 different years. 6 seed treatments were used, and I'll show you what those were in a moment. So what they did then, they took nematode populations at planting and later in the season, in all those treatments. Then they looked at the number of nematodes in plants, in roots at 30 to 35 days after planting. I'm going to show you numbers in a moment, but here's the overall conclusion. No seed treatment across all these locations, when all the data were pooled, offered significant SCN control versus a non-treated check. Of all of them, Alivo and Saltro provided more consistent increases in yield. I suspect that is primarily due to their very strong effect on SDS. Now, this shows data. Now, there's a lot more data in this paper, but just to answer this question that came up, what about comparing those two products and how they affect nematodes? And so I don't need to read all this. If we just look at this table in the middle, it shows the treatments here, all the different seed treatments that were used, the non-treated check at the bottom. And the 2 columns of data show females this is SCN, per gram of root. This is 30 to 35 days after planting. And the next one is a reproductive factor. Let's get to that in a moment. So again, the number of nematodes per gram of root for the non-treated at the bottom, the very bottom treatment was 6.2. And what we want, of course, is to see a treatment that reduces the nematode numbers. What you see here is only one of them really did. And that was Alivo, although it was not statistically significant. It reduced it from 6.2 to 5.4. All the others, most of them had no significant effect, in fact, at all. Most of them did not. Okay, for the reproductive factor, there's no statistics attached to this, but it shows the numbers. Again, it shows we would like the number to be lower, right? Lower than the non-treated of 6.7, and most of them were lower, with a few exceptions. And again, that's the population at the beginning of the season at planting compared to the end. So again, they did not see a strong effect of these seed treatments in this paper on SCN. That was the upshot. The last disease, main disease I want to mention here in terms of emerging, and I think this is likely truly emerging one. If we continue to have periods of drought and heat in the summer, I expect this is a disease we'll see more of. It's very common across the US. We know it's in Minnesota, and we don't know how widespread. I think it's really causing problems yet. I can tell you that right here on the St. Paul campus, there was a field with soybeans dying, in July, and I went out there to look at it, and it was charcoal rot. It was very, very dry there and fairly warm. So it's favored by these very hot soil temperatures and very dry soil. Usually is most commonly seen late in the season after stress on the plants. And unfortunately, this has a very wide host range, this charcoal rot pathogen, including corn, alfalfa, sunflower, dry edible beans, and other things. So the symptoms, again, it usually begins in the driest parts of the field, premature yellowing of leaves. And these are the kind of the diagnostic sign. If you scrape off essentially the lower part of the stem, the root, or cut into it, you can see these black spots. Now, I'm not sure how clear this image is showing up, and in the middle of this, you can see black spots that look like specks of pepper. And they can be seen with the naked eye, but they're certainly much easier to see with a hand lens. But they are big enough to see with a naked eye at times. But don't count on that. Just use a hand lens, and these things show up very clearly when charcoal rot is there and causing significant damage. One thing to keep in mind, there's another disease out there, pot and stem blight, that can cause some similar type symptoms from a distance. But we typically would see these black specks on the outer side of the stem in rows. Now, these are much bigger than the charcoal rot spots. And this also is typically, if you cut into a root and you see gray swirling lines in the root, that's more often associated with pot and stem blight than charcoal rot. So that is not necessarily a diagnostic feature of either one. And again, looking at these stems, these were initially thought to be charcoal rot and turned out to be pot and stem blight. So this is one we really want to know about. And so if you're out and about and see samples that you suspect could be charcoal rot, we like to get samples to document when and where this disease is occurring. We think it may be widespread. And what about managing it? When we have an issue, rotation with small grains may be helpful because they are not susceptible to charcoal rot. And there are varieties with some level of partial resistance. Now, looking ahead, a big part of this every year, obviously, is our weather. Hot and dry, we get diseases like charcoal rot. Wet and cool, we get diseases like white mold. We never quite know what we're going to find and what's going to develop. We can look ahead, we can look back, we can learn a lot from what we see, but the key point is everybody keep their eyes open like always and look for new things, because it's not a constant out there, as we all know. And with that, I have the end of my slides, so if there are any questions, be glad to take any.
Thanks, Dean. Yeah, thanks. There are several questions, so I'll get those pulled up here. But there were a few questions about tar spot, so I'll kind of hit you with a couple of them here. One participant says they've read in different articles that tar spot is triggered mainly by humidity levels of 75% and higher for 10 consecutive days. And they're wondering if you agree with that.
Well, not too much higher. I think it's the range that we're looking at, it seems like, is somewhere between 75 to 90. If we go a lot higher, that doesn't seem to really support it, at least based on some of that information. And again, we saw a significant tar spot spread in our plots when we had low humidity, even below 75 at times, although it did reach 75 sometimes. So it's not absolutely clear. And the main point is we don't need real high humidity.
All right, thank you. And somebody is wondering, are there any more sensitive crops, maybe like sweet corn, that could serve as a sentinel for trying to detect tar spot at the low levels?
Yeah, from what I've seen, tar spot isn't any more sensitive than a susceptible dent corn hybrid. Maybe somebody out there has seen something different. Certainly it develops in sweet corn, but many dent corn hybrids are also very susceptible.
Okay, so Yeah, so nothing really that can be your indicator before it hits
No,
your field corn.
no.
Yeah,
I think the key is just have a corn hybrid out there that is very susceptible,
sure.
whether
Yep.
whichever type it is. Yeah.
And then this question is more about your and Jose's research methods, but the question is, what's your inoculation process, your field inoculation process with tar spot?
Yeah, very, very simply, Jose takes infected corn leaves with lots of tar spots on them, and he grinds them, suspends them in a water solution and sprays them on the plants. It's that those are the simple steps. There's a little more to it, but that's the basic method that's used. He did a lot of trial and error work to figure out some of the details within that, but that's what's working at this point. Yeah. And a key thing that he found is that if we keep tar spot infected leaves, say on the lab, and let them dry, keep them at 70 to 75 Fahrenheit in a room. If we keep them over winter, we can't get infection with them. If we freeze them, then they stay viable and we can get infection.
Interesting, yeah. This question is kind of more general in regard to all the diseases you mentioned, I think. A participant is wondering if this information can be found on our extension website. And they give a little context that they're looking to create a sort of disease compendium for themselves for reference throughout their career and would like to get these sorts of things down in detail. And maybe you can talk about some of our resources where people can find information on this.
Yes, so I have some information, especially on the soybean diseases, on a website at the University of Minnesota. There are many resources. There are resources on, I think, every one of these diseases on the Crop Protection Network website too, including publications that have extensive description of diseases and symptoms, and the conditions that favor them for almost all the diseases that I talked about, and maybe all of them. And those are written by a group of people across the Midwest. And so they're vetted and sort of agreed upon by a lot of folks. So if you want to know about white mold, there are several publications there about white mold, sudden death syndrome, tar spot, etc. So that's a good place. And people from all across, colleagues of mine, plant pathologists from all across the US are contributing to these. And that includes me. And we all work on developing these, so they're useful for everyone. So take a look at that. And again, our website here at the Minnesota has some information, not as extensive. And if there's anything you're looking for, please don't hesitate to contact me.
Yeah, thank you. And we will include links to those resources in our kind of follow-up resource document as well. And this question came up when you were talking about soybean cyst nematode. What are your thoughts on rotation of genetic tolerances? And this person gave an example of peaking with like PI 88788.
Yeah, I think it's the right thing to do if it's feasible. Yeah, and when I say it's feasible, we're having more and more varieties of peaking right now that have good yield potential, have good resistance, and so that's generally considered a very good thing to do. The danger, of course, let's see, I'll say start there. That is the recommended practice, to rotate sources of resistance if it works in our system. And there's more and more varieties, like I say, where it can work. But we have to keep in mind that peaking resistance is not invulnerable either, because definitely nematodes can develop abilities to overcome that resistance too. So something to keep in mind.
Thank you. And this next question has a product that I'm not sure how to pronounce, so you may have to correct me, Dean, but does a strobilurin fungicide result in more green tissue at harvest? And if that's a concern, would a triazole or SDHI be better for late season application?
Yes. Strobilurins now more commonly called the QOI fungicides, so there's the same thing, can cause late season greening and increased moisture content in corn. That's widely known. It doesn't always occur, but certainly we see it. And we can see it sometimes in soybean too. Triazoles are not as associated with that. The strobilurins seem to have a greater ability to have that stay green effect, although the others can have some effect too.
Thank you. Yeah, and the SDHIs at all? Any thoughts on those? Or is that...
they can do it a little bit, but again, from what I've seen, the strobilurins have the greatest effect on the stay green issue. Yeah.
All right. And do you think earlier planting dates are associated with greater incidence of crop disease in corn and beans?
In some cases, yes. In some cases, no. That sounds like a cop-out answer, doesn't it? But it really does matter. A lot of it's driven by the weather, right? Because if you have early planting for soybeans, it might make it worse for SDS, for example, when you have very wet soil. Whereas if you plant later in May and then it turns drier, and it's not very moist in the soil system after those seeds are in the ground and they start to germinate, then you may have less SDS because the moisture at the time that seed germinates is important for that initial infection. For something like tar spot, that's a disease that develops very late. So if we can get a crop that matures earlier, and we don't need to lose yield in it, and that's the challenge, we can maybe avoid some of that typical late season increase in tar spot when that disease tends to really ramp up in late August and early September. If a lot of the crop is already made by then, we can reduce yield, I would suspect. So it varies a lot from disease to disease and partly based on the weather each year.
Yep. Well, you didn't say it depends, you just said it varies. So that's
okay.
And how are there any pros or cons to using a drone to apply fungicides? We know that's becoming a little more common.
That's a great question. And certainly one of the pros is the ability to get into smaller areas that might be protected by trees or power lines or other kind of obstacles. I think we're learning a lot there. Drones can really have a place. I think the applications can be effective from what I've seen. I think we have a lot to learn. But I'm optimistic that they could be very beneficial for many situations.
Yeah. Yeah. A question about how often should a grower use peaking resistance for SCN, soybean cyst nematode? Is every third year of growing soybeans adequate?
So assuming it's a corn soybean rotation, I would actually consider putting it in every other year. There's no definite answer to that one, I think. Maybe some might argue use two different varieties of 88, 70, 88, and then use a peaking if you really want to minimize the chance that peaking will lose its resistance. Some would say maybe rotate every other year.
Yeah. No, thanks for your thoughts on that. Another question is how severe is brown stem rot of soybeans in Minnesota?
That's a good question. That's one we've done a lot of work on. It can be very severe. The brown stem rot is another fungal disease, and it's quite widespread. We don't see it at high levels every year, for sure. But in our field trials in Waseca and Rosemount the last few years, it's been very common. And, as a reminder, we don't always see the leaf symptoms. Sometimes you actually have to split the stem to see if brown stem rot is there. And the question might come, what's it doing to yields? That's a good question. Now, in a very susceptible soybean with high levels of brown stem rot, it can approach 30% yield loss. I suspect we rarely see that anymore, Most varieties have a reasonable level or a high level of brown stem rot resistance, although we still see brown stem rot developing even in those in our research plots. So I guess that the thing is, I think this is an important disease, maybe more so than we recognize in many cases.
Are you seeing, I don't know if you have this information, but are you seeing yield impacts in your test fields too that have resistant varieties?
You know, that was hard to say because we didn't have a comparison. Everything had brown stem rot.
Oh, sure.
But when we've done, one thing we've done, when we haven't even seen a lot of foliar symptoms, we've measured about 12 to 15% loss a few times. So I think that sort of loss is much more common than that higher, above 20% loss. But sometimes it's kind of hard to measure because when it develops pretty broadly, and it's hard to have side-by-side comparisons.
Sure. Yeah, well, we have time for more questions, so I'll just ask our participants to put any burning questions you have in the Q&A, or we'll pull questions and keep going there. But in the meantime, I'd ask Dean if you'd like to talk a little bit about the digital crop doc we have with Extension and how folks can submit samples to us in Extension.
That's a great question. I'm glad you brought that up, Claire. The digital crop doc is something it's a website, essentially. You can submit samples. Well, put it this way, you can submit photographs of symptoms on that, and they'll be sent to a central location, distributed experts all across the University of Minnesota system to identify what the problem is. So if you have a leaf disease, you want to know if it's X, Y, or Z, put a picture on there, provide a little information, it'll go in, and somebody will provide a diagnosis for you very quickly. And if we can't give a definitive diagnosis, with a photograph, which is often the case, we would suggest you send a sample to the plant disease clinic at the University of Minnesota. But often we can provide, some diagnostic guidance, even based on the photograph, if we have good photographs. So you can look up that system, and we'd welcome anybody to use it because we think it could be very helpful for many folks.
Yeah, well, thank you. Yeah, and I think you said it's free of charge, and it's, I think, a great tool too. So a question just came in on how do cover crops help or hinder crop disease?
Yeah, that's the I had that question. I think it depends on again, that depends word. The cover crop and the situation. I've seen results both ways. There's some information out of Iowa that some cover crops can actually increase, say, the Pythium population in soil. And Pythium is a soil-borne pathogen that can infect soybeans, corn, and almost anything, any crop. Now, whether it's enough of an increase to have any substantial effect, I don't know. But it certainly, some of those are susceptible to some of the pathogens and can increase the population. Now, does that increase disease and ultimately reduce yields? I think we have a lot more to learn on that. I don't think we have enough information to provide any definitive answer. On the other side, we know that cover crops can increase the microbial diversity, which can also be suppressive to some pathogens. So I think we have a lot to learn on that one still.
Yeah, agreed. There's some, I think, work being done on looking at like predatory nematodes being present with cover crops, things like that too. But that just kind of made me think about, kind of on the soil health track, how does maybe reduced tillage help or hinder crop disease? And I kind of bring this up because we get that question with corn tar spot in particular, because it overwinters in the residue. So how does residue management and tillage maybe affect that disease or other diseases?
Yeah. So I think I can group that. Let's just, if we think about it in terms of corn disease, you mentioned tar spot, Claire. Residue management hasn't made a lot of difference in the trials that have been done, partly because the pathogen can be windborne, can be spread by wind. I think we might get some earlier season infection in some fields if they're heavily infested with residue and it's right there in that field. But at the end of the season, sometimes the differences aren't very clear. If it's been, really plowed and buried versus not really managed very much. Whereas I mentioned anthracnose before. Now, this wasn't not direct field research studies, but somebody did a survey across multiple fields. This was like 20 years ago, showing those fields where no-till was used had a higher amount of anthracnose on plants. So corn anthracnose was more common in no-till field situations. Yeah, that is a disease that probably isn't so prone to being spread by the wind. And some other diseases like white mold and SDS, the effects of tillage and residue management are not so clear. You see results both directions.
Oh, interesting. Yeah. Yeah, thanks. Yeah, a question just popped in. Good. Oh, just wondering about the continuing ed units. Yep, those will be at the very end. We'll have a QR code come up on the screen and you can use your app to get the CEUs. And I think we have time for a little bit more. If you have any thoughts, Dean, or maybe you could tell folks where they could go for updates, or maybe to keep track of emerging disease issues as they're happening, or track the spread of various pests. Do you have any resources for that?
Yeah, the one thing I'll kind of repeat is I mentioned the corn IPM pipe website for tar spot. So you can see updates throughout the season where that disease is being found in the US, including Minnesota. And that also shows updates for a few other diseases. And one is southern rust of corn. That one's also kept track of. That typically truly was a more southern disease, but it's been developing more and more in the northern US, including in Minnesota at times. And I briefly mentioned this before. There's a disease that is of concern because it can really take off later in the season when it's too warm for common rust of corn. So again, the corn IPM pipe website shows information where that disease has been reported and found in each season too. But for some of the other diseases, there is no central reporting point to say where and when they are developing.
Yeah, thank you. Yeah, good mention of that resource again. I see somebody's raised their hand. I don't know if we do anything with that. Oh, and then he asked the question, yeah, where do we go for disease updates during the year? So I think kind of that IPM pipe is that good resource for that.
Yeah, for a limited number of diseases though. That's
right.
It's only limitation right now. Yeah.
Thank you. Yeah, do you have any parting thoughts for us, Dean? Otherwise, I'll kind of give us our wrap-up spiel here and we can get people their CEUs.
Yeah, I don't have any big points, but I just a reminder that if anybody has suspected charcoal rot samples, we'd like to see them. I didn't mention Phytophthora today, but we're also looking for samples of Phytophthora or fields that are infected with Phytophthora to try to figure out what's the race pathotype is out there across the state. So there's no charge for any of this analysis. It's part of some research projects. So we'd welcome any plants or soil from fields that are infested and problematic for those two diseases especially. They can be just sent to my attention at the University of Minnesota.
All right, well, thank you. Thanks for that and those reminders. And thank you all again for attending the Strategic Farming Let's Talk Crops program today. And I want to say thank you again to our sponsors, the Minnesota Soybean Research and Promotion Council and the Minnesota Corn Research and Promotion Council. And this is the last session of our winter series. We'll be back at it with our field notes series starting later this spring and then running throughout the growing season. So stay tuned for more information on that. And thanks again.